Hi, so Tuesday afternoon, 5 o'clock, it is time for us to go through a biology paper. So let's hope that I have got this right. Okay, and we are going through the AQA A-Level Biology Paper 3 from 2018. So without further ado, I'm going to make a move and get started. Probably just before I do that, I should probably say to those of you out in video land, you should be ready to stop the video when you come to a new question. I'm going to really briefly go through what the question is saying. I'm then going to talk about how I would have answered the question, show you the mark scheme and the additional notes from the examiners, and then show you where I would have picked up my marks. Really precisely because I mark exactly where I think I gained the mark point and number each of those mark points according to the numbers that are in the mark scheme. So hopefully this now makes sense. And welcome, can I just say, to those of you who are watching now, well done. So section A. This is, I'm going to focus on the diagram here, so to guide me through the blurb at the top. And the blurb at the top is basically saying that an osteoblast, which is a cell in the bone, produces inactive osteocalcin. But when there is acid around, the osteocalcin becomes active, and that stimulates B cells to produce insulin that in turn stimulate the osteoblast. And here we have used the information from figure 1 to explain why this is positive feedback. Well, that is because the production of the active osteocalcin stimulates the beta cells to produce insulin, which then stimulates the osteoblast to produce more osteocalcin, which is pretty much what I have written here. And at the end of this answer, I wasn't even sure that I'd written quite enough. So I actually kind of gave that definition of positive feedback to really show that I knew what the content was. So here's the mark scheme. Here are the notes. I don't think I need to use the notes on this. And therefore, this is where I got my mark. Now, notice that I didn't get the mark. I didn't get mark 0.1 because I didn't say that it caused the release of more insulin. And that was clearly a very important part of the answer. I just said that it causes it to secrete insulin. So I actually missed the first mark. Here. The acidic pH conditions created by the osteoclast cause the inactive form of the protein osteocalcin to change into the active form. Suggest how. So I basically said that if there was an increase in hydrogen ions, I went back to my protein stroke enzyme knowledge, said that an increase in hydrogen ions would break those ionic or hydrogen bonds and hydrogen bonds, because that equilibrium of hydrogen ions has been disrupted. And that could therefore change the tertiary structure and of the binding site, which allows the osteocalcin to bind to the beta cells, stimulating the release of insulin. And this was the mark points. So I got both the mark points on that question. Hello, Dr. Cheddar Cheese. Loving that name. Okay, on to question 1.3. Binding of insulin leads to an increase in the rate of respiration. Explain how. So I just want to point out here that the binding of the insulin is kind of where the start of your answer needs to happen. And you have to finish your answer relating it to an increase in respiration. So here I talked about, not surprisingly, that insulin binding causes more glucose channels to be added to the osteoblast. And you learn that in your regulation of blood glucose. You should have learned that insulin increases the number of glucose channels in body cells generally. And therefore, this increases the uptake of glucose, and glucose is required for respiration. I just put, and therefore an increase in respiration. So I got both of the marks there. This being the mark scheme down here. This on this side of the screen, I've put the examiner's points that help examiners mark, or the chief examiner's points that help the examiners mark the paper. Okay, this is about chronic lymphocytic leukemia, CLL, which affects B cells of the immune system. And rituximab is a drug that treats CLL, and it does that because, and here am I, I've drawn myself a little diagram, as it were, in the margin of the paper. Here's rituximab. It binds to a receptor, or the CD20 receptor, on the B cell. And if there is enough rituximab, it causes the death of the B cell. But it causes the death of both healthy and cancerous B cells. Healthy being sad and cancerous being good. But then the body makes more. But I've just tried to encapsulate this blurb in a silly little diagram just to get the crucial points into my head. But of course, the first question has nothing to do with understanding the blurb. It's asking about the type of statistical test. Now, here you have got two variables that we are seeing whether there is a correlation between them. The amount of CD20 on the surface of B cells and the percentage of B cells destroyed by rituximab. And so the answer is, I've just put Spearman's rank, and I've said it's because we're looking for the degree of correlation between two variables. You could have said between two dependent variables, because they are two variables that have had to have been measured. And dependent variables are variables that you measure in an experiment. Oh, Harriet, how do you recommend dealing with those really wordy questions? So I would always say, if you are looking at trying to write an answer for a 3 or 4 mark question, particularly for AQA, I would say, number one, don't be put off by the number of lines they give you. If you end up finishing your answer halfway down the number of lines, don't feel like you have to fill the lines. AQA particularly are now giving a huge number of lines for each answer. The other thing I would say, read the question carefully and try and stick to answering the question. From a very practical point of view, I would also say, try and keep your sentences short, because try and make one statement and then follow it by your next statement. I have so often seen where one answer is basically one sentence, which is about 6 lines long. And actually, that kind of writing is very difficult to follow, potentially. And also, it's far more likely that students will get their its and theys written in a way that the examiner doesn't actually know what you are talking about. So I would personally say, read the question, try and stick to answering the question, keep your sentences short, and also I would always say, even if you don't completely understand the question, use the terminology for that topic. Okay, so, and I will talk specifically about some types of questions. So, for example, let me just do it now, as soon as we're talking about it. If it were an explain question, explain this data, for example. And I'm looking at the data and it's a 4 mark question. And I'm looking at the data and I'm kind of going, I could answer why that graph is the shape that it is in kind of one sentence, and I call that my headline answer. I fundamentally know what the answer is, and I'm kind of going, great, that's given me 1 mark. What about the other 3? And here's my clue. If there isn't anything else obvious that you can write to explain the answer, and you're kind of going, I don't know how else to get the marks, I would then fall back on using my A-level biology content knowledge to add detail. It's almost, even though it's an explain question, you are then using content description, or maybe I should say description of your A-level content to fully describe the biology behind your headline answer. And because I right now I can't think of an example, I'm sure we'll come across one. So if we do, I'll point that out. I hope that helps, Harriet. I hope that gives you some pointers anyway. Let's move on. Oh, so yes, the notes here say that they're allowed to accept either Pearson's or Spearman rank. So that was fine. So from these data, what can you conclude about the effectiveness of rituximab in treating patients with this type of cancer? This is an AO3 question because it's asking you what you can conclude from the data. And this is not a surprise in the paper 3. Most of the paper 3 marks are either AO2 questions, i.e. applying your knowledge, or AO3, which is about evaluating, drawing conclusions, interpreting data. There are relatively few AO1 marks, and ironically, they're in the essay at the end. We'll come to that later. But worth knowing that this paper 3 is going to feel harder. Hope that makes sense. Okay. What can you conclude? And it tells you not to include considerations of statistical analysis. They don't want you talking about stats. So what can we conclude about the effectiveness of rituximab? So here I would say that the effectiveness of rituximab increases as the number of CD20 receptors on the cells increase, and here I've told the examiner that I'm using the initial R, or the abbreviation R, for that long drug name. Quite useful to do that. So here I go. The effectiveness of R is determined by the number of CD20 receptors. The higher the number, the more effective it is. Thereby, I've given the relationship. And I'm kind of going, okay, that's my really obvious statement. And I'm looking at this graph and I'm going, okay, what else can I say? And I'm noticing down here on the graph that actually the amount of CD20 only make a difference when you are above a certain number. So, and therefore that's my second point. However, there is little effect on the amount. If the amount of CD20 on B cells is below approximately, and I looked at this quite carefully, 4.5 arbitrary units, AU. And then I noticed, so here am I, and then I'm kind of going, okay, let's just take a step back. This is about treating cancer. Ideally, we want to kill off every flipping cancer cell. And here we go, noticing that actually at the top here, we've actually got only about 80%, here am I looking at the graph carefully, 76% of the cells are destroyed. And as I was writing this sentence here, it suddenly dawned on me that actually in my little diagram that I put next to the blurb of the top of the question, that I'd noticed that actually the cells will consist of both healthy and cancerous cells. And so and because this is about treating patients with CLL, getting rid of the cancer, and therefore I just noted that 76% of cells was both healthy and cancerous, and therefore the proportion of cancerous cells is unknown. So, and here is the mark scheme, here are the notes, and here are the marks that I got from the mark scheme. So I ended up picking up 4 marks, but of course there was only a maximum tariff of 3 on that question. So here it says approximately 11% of people with this cancer also have mutations of a gene called Notch1. This Notch1 gene leads to the production of a non-functioning transcription factor. Here I am, scrawling notes in the margin. N1 mutation leads to transcription factor not working, which I have just kind of said, well, that must lead to a decrease in the amount of CD20 receptors produced. Because if there is less transcription factor, at AQA A level, you fundamentally learn that the binding of a transcription factor initiates transcription. Therefore, if it's not working, less transcription, less protein. And here I've pointed out, so here is the table. So in people with cancer who have had the Notch1 mutation, this is, and for the moment I'm going to disregard the median, you know, the percentage of B cell destroyed is only 4%, far less destroyed with the mutation than without. And that makes sense. And that must be because there is no or very little CD20 receptor for the rituximab, I've now forgotten, to bind to. So I've read through the question, I've kind of got my head around what it's talking about. And then, of course, the first question is about something completely different. Hi, what? And here we have got to use the median values to calculate the difference between the number of B cells per decimeter cubed in the blood of people treated with rituximab and those without. So I worked out, so I know I'm just going to show you the way my mind works. It is not always the simplest, but here am I kind of going, okay, they've got, they go down to 96% of their B cells, and these go down to 78%. That is an 18% difference. Clearly, it is the same percentage difference between these two numbers. I'm just showing you the way my mind works, all right? Because I'm not that intelligent, all right? So, you know, so here we go. So we have been told, now where have I got? Oh, yes, so 0.18 is, of course, 18% as a decimal. So it is this proportion of this number here, because they tell you that normally this is how many B cells there are per decimeter cubed in normal blood. So therefore, it is, if it's 0.18 times 1 times 10 to the 9, it is 0.18 times 10 to the 9, which equals 1.8 times 10 to the 8. Doesn't have to be times quite so many times by 10, if that makes sense. And therefore, and they tell you to put your answer in standard form and show you're working. Maybe I should have put either this number or 22 minus 4 equals 18 up here as well. So, and therefore, because I got the right answer, you just automatically get the marks, regardless of anything else. This kind of surprises me, that if somebody uses 10 to the minus 8, that you'd even get 1 mark. I'm going to be really honest about that, because 1.8 times 10 to the minus 8 means 1 over 1.8 times 10 to the minus 8. A way tinier number. I hope you're appreciating. Anyway, moving on. Oh, here we go. This might be a bit of a blurby question, Harriet. So use all the information to suggest how the mutation of Notch1 led to the difference in the percentage of B cells destroyed by rituximab. So here we go. So this is using, let me just go back, this is using all of the knowledge. So the fact that we're talking about a mutation, and we're just, and also understanding how rituximab works. So right back here to my silly little diagram at the top here. So it's really taking on board everything that you have learned, or you've been told rather, maybe I should say that. Okay, so here am I. Here is the start of your answer. A mutation of Notch1 led to a difference in the percentage of B cells destroyed by rituximab. So this is the start of your answer, and this is the end. You've got to start by kind of going, well, if there's a mutation, then this is going to happen. And work your way through to the end of the scenario, which is the percentage of B cells destroyed. So hence my answer. Mutation of the gene means that the order of the bases in the DNA is changed. It could lead to a change in the primary structure of the transcription factor. Notice that I have shown my understanding of how the Notch1 works. And therefore, the tertiary structure of its DNA binding site, because that is the crucial thing. That transcription factor has to bind to the DNA, causing it not, and I've tried to capitalize this, not to be able to bind to the DNA. This would stop or decrease the transcription of the CD20 gene, leading to less CD20 receptors being made or synthesized. Less CD20 receptors lead to a reduced effectiveness of rituximab because of the reduced binding with CD20 receptors. And therefore, I could have put a full stop here, that might have been good. And therefore, and here am I finishing off, clinching the deal, hopefully, and therefore a reduced percentage of B cells destroyed. I've gone from the start to the end. This isn't like the example that I said to you earlier, Harriet, where it's kind of explain and, where the rest of the marks. This is about realizing what the start of the question is and realizing what the end is. And here's the mark scheme. Here's the extra comments. And this is where I got the marks. And I think, yeah, I did end up getting 4 marks, but of course I'd have only got 3 in the mark scheme. I should just say that when I wrote the answers to this paper, you and I both know that I have made this PowerPoint, and therefore I was cutting and pasting mark scheme answers and comments onto this PowerPoint. I am just going to tell you that when I came to actually, because I do that all in one process, and then I go through the questions and I write my answers, and I deliberately don't look at the mark scheme on the left, on either side. And I know you're not really going to believe me, but, hey ho, that's what I've done. Because I want you to see what I would answer, because I'm not completely sure what level of detail is in the mark scheme. And we all know sometimes it's a one-line answer, and sometimes it's the last bit that you put on the end of a sentence that actually clinches the mark. And I'm afraid I don't know exactly what is going to be on the mark scheme. I might have a pretty good idea, but exactly what's going to clinch the mark point, I don't know. So anyway, that might have given you time to have read this question. This is about how eggs are actually produced in meiosis. You have not learnt this. It is not part of the AQA syllabus. So you are expected to take all of this information on board. Basically, a bog standard ovary cell will undergo meiosis, and what actually happens, the first meiotic division, you get one normal egg and you get what's called a polar body. The polar body doesn't carry on dividing, only the normal egg does, which then goes through its second meiotic division. But then, of course, it's only going to split into two cells, and therefore you get finally from meiosis to make eggs in women, you actually don't get 4 eggs, you only get 1. And here it is. Here is the final normal egg with 2 polar bodies, and only 2 polar bodies because, of course, this first polar body didn't go on and divide to produce another one. I hope that makes sense. And here is the question. So once you've got your head around all of that, here is the question. Complete figure 3 by putting big A or little a in the boxes. One box has been completed for you. Well, thank you very much. Okay, here is our bog standard cell showing 2 homologous chromosomes. One chromosome carries the allele capital A, one carries the allele little a. Now, you have to notice here that actually crossing over has occurred. There has been an exchange of alleles or exchange of part of non-sister chromatids during that first meiotic division. And therefore, this is little a. Excuse me, sorry, because each chromosome has now ended up with one big A, one little a allele. And now we need to look carefully at this second polar body. So in this second polar body, this one has got the big A chromatid in it. So therefore, this one must be the little a one. And therefore, that's why that is correct. Put a tick in the box. The name of the process producing a combination that produced the combination of alleles on the chromosome in the first polar body. Well, that is crossing over, which is what I've just talked about. So we've got that mark. And now here, let me just check. Right, now they're talking about the polar bodies, and you'd been told that the polar body, or you're told that the diameter of the nucleus in the polar body, which they've kind of indicated here diagrammatically, is really quite big in comparison to the diameter of the whole cell. So they tell you that the diameter of the polar body is 10.4 micrometers and the diameter of the nucleus is 7. And they also tell you the density, the number of mitochondria packed into a specific volume. The density of the mitochondria in the cytoplasm of the polar body is 0.8 mitochondria per micrometer cubed. So calculate the number of mitochondria in the polar body. So what we have got to work out, because of course those mitochondria are in the cytoplasm, which they very kindly tell you, you have got to work out the volume of the cytoplasm. In other words, the volume of the polar body minus the volume of the nucleus. So here we go. I'm showing you the long-winded way first. So here is and notice that of course this is diameter, not radius. So you've got to, use half of D. So here is the volume of the whole cell. Here is the volume of the nucleus, and you take one away from the other. I've put a recurring sign over the 3 here because in my calculator, it carried on. And I would always keep the whole number in the calculator and use that whole number until I was going to write my answer on the line here. I happened not to do it here. And notice, so this number here is the number of micrometers cubed of cytoplasm. And you are told that for every micrometer cubed of cytoplasm, there are 0.08 mitochondria. So does it make sense to work out the number of mitochondria? You times those two together to get this number here. And I have rounded this up because they're asking you, how many mitochondria? Well, you can't have 0.7 of a mitochondria. You've got to have a whole number. So I've rounded it up to 33. The mark scheme absolutely allowed you not to do that. And therefore I got the 2 marks there. Now, here is the faster way to do it. Because, of course, the only thing in these 2 sets of calculations that change is the radius. And therefore, if you work out the radius cubed and take one away from the other, and then times that by the rest of the calculation, you end up with exactly the same number, and potentially is quicker. That's absolutely fine if your mathematic ability completely helps you to get that and just gives you a shortcut. If there is any question of your maths ability, just do it long-handed, because that's a nice 2 marks. Okay, so we have, mitochondrial diseases, they talk about here, it talks about how they are kind of cured. You take the nucleus out of a healthy woman's egg, you put in the nucleus of the egg of a woman with mitochondrial disease, you can fuse those together, and then the woman with mitochondrial disease can go on to have children who do not inherit that mitochondrial disease. And children will inherit that mitochondrial disease because we have inherited all of our mitochondria from our mother. How weird is that? None from your father, because the only bit that he contributes is the nucleus of the sperm. None of the cytoplasm, because basically there's hardly any cytoplasm in the sperm, is there? So anyway, right. Suggest how this treatment prevents the inheritance of mitochondrial diseases. And I'm kind of going, crumbs, this seems kind of quite obvious to me. Wasn't quite sure how to nail it. So I kind of said, this is what I put, because the faulty mitochondria are not in the egg cell that are fertilized. The egg to be fertilized has the donated cytoplasm with healthy mitochondria, and it has the nucleus containing the DNA from the woman with the mitochondrial disease. So here's the mark points, and this is where I got my marks. Notice first 3 lines didn't earn me anything. Anyway, moving on. If most of the mitochondria in the cell are faulty, this prevents many important enzyme catalyzed reactions taking place or slows them down. So here we have got to make the link between mitochondria and enzyme catalyzed reactions. And that is ATP. So, and because it's mitochondria, I nearly just wrote here because less respiration takes place. But as I was writing because less, I realized, okay, we're talking mitochondria. I know we're talking aerobic. I'm going to put the word in. All right. Takes place and therefore less ATP is produced. ATP hydrolysis, and just as I always just put this in because it takes me no time at all. Sometimes it is an extra mark, and it provides the energy for many enzyme catalyzed reactions. And then I kind of went, how does that? And then I remembered, now this is me remembering from glycolysis in respiration, the transition of glucose to phosphorylated glucose or glucose bisphosphate, or I can't remember the other terms. Basically, what happens, the ATP has provided the phosphate by its hydrolysis that has caused the phosphorylation of the glucose. And I know that is why the reaction happens more readily, because that phosphorylated glucose is more reactive. So I put that in. Here's the mark scheme, and here's the extra points. So I actually ended up getting both the marks, actually. Oh no, this was OR. Yeah, I ended up putting in both of these, but I only got 1 mark, obviously. Okay, really quick plug just for you out in YouTube land, as it were, or video land, not in live stream. Just to know that I have got a website with the full complement of videos that cover AQA and OCRA syllabuses. That means it will cover a huge amount of content for every A-level syllabus. Each video covers part of the syllabus. If you want to subscribe to all the videos, I'm still doing this at half price. It's not £100 the whole year, it's 50 quid, which is fundamentally 1 hour of tuition. AS videos, just those first year, it's 25 quid. For those of you, this is my great effort to make A-level biology accessible to every student, regardless of family income. You can earn your subscription back. If you recommend it to your friends, you get 10% commission. And there's a freebie on there, how to improve your grade up to an A star, that gives you real tips as to how to just pick your grade up, regardless of what grade you're aiming for. So do have a look. Anyway, back to question 4. Binding of one molecule of oxygen to hemoglobin makes it easier for a second oxygen molecule to bind. Explain why. Well, that's because the binding of the first oxygen molecule causes a change in the tertiary structure of the hemoglobin, which kind of opens out the second oxygen's binding site, and therefore makes it easier for the oxygen to bind. So, because the binding of the first oxygen molecule causes a change in the tertiary structure, making it more accessible to the oxygen molecule. It could have said more open. I wasn't quite sure whether this word was good enough, but just went for it. So I got the mark point here, and I think I got this second mark as well. I'm pretty sure. Creates leads to uncovers second binding site. I think I've got that. Hemocytometer. Special microscope slide. It's a 3D microscope slide. It holds a volume, a very small volume of blood. It's thin enough, the depth of this cuboid is thin enough, obviously, to allow light to pass through it so that scientists can count the number of red blood cells. And it's, this is like a quadrat, isn't it? This is, just like a quadrat, but which has got a volume. And you count the number of red blood cells in each known volume of blood, notice. The doctor counted, the mean number of red blood cells per small square was 7. The original blood sample was diluted by a factor of 200. Calculate the number of red blood cells per millimeter cubed. Okay, first of all, we have got to work out the volume of this cuboid. 0.05 squared times by 0.1. So there are 7 red blood cells in this volume. 7 red blood cells in 2.5 times 10 to the minus 4, because on my calculator it was 0.0003025 in that volume of blood. So therefore, in one volume of blood, I've got to scale this number up, scale this 7 number up. And so therefore, I've got to know how many 2.5s times 10 to the minus 4 go into a whole 1 millimeter cubed of blood. So therefore it is 1 divided by 2.5 times 10 to the minus 4. And then I've got to times it by 7 because there's 7 in this volume, which gives me 28,000. But I haven't yet taken into account that's in the diluted blood, and it's been diluted by a factor of 200. So I need to times this number by 200, which gives me 5.6 million. But look, give your answer in standard form. So it is 5.6 times 10 to the 6, because 1, 2, 3, 4, 5, 6 decimal, the comma needs to be moved 6 times. Okay, hopefully that makes sense. When counting, cells that touch the top or left lines are counted, but cells that touch the other lines are not. Suggest 2 reasons. So I almost have a knee-jerk reaction here. And my first response was, this is a standardized method so that everybody is doing the same thing. And it also very obviously makes sure that cells aren't counted twice. So hopefully that makes sense. Oh, sorry, those are my marking. So here we go. Doctor also wanted to know how many white blood cells per millimeter cubed there were in a different sample. He diluted a sample by a factor of 20. Oh, different to red blood cells. He then made the white blood cells clearly visible by using a stain that stains the nucleus. It suggests why he only diluted the sample by a factor of 20 times. And I just put, because there are far fewer white blood, oh dear, I don't know where the D went, blood cells than red blood cells. In fact, there's approximately a 10th less because the dilution is a 10th less. And I kind of thought that would be enough for 1 mark. But look, I didn't get the mark because there was no need to dilute further. I kind of thought that was self-explanatory from the question. But anyway, so I didn't get the mark. Explain how the stain allowed the doctor to count the white blood cells. Well, that's because the stain stained the nuclei. White blood cells have a nuclei, have a nucleus, red blood cells don't. And also, I'm also thinking, well, red blood cells are red. White blood cells, quite possible that they would be very difficult or you couldn't see them in that kind of scenario of looking down a microscope, and therefore staining was needed to see them. So I put, because only white blood cells have a nucleus and they wouldn't be seen clearly without staining, unlike red blood cells. And I did think it was a little bit patronizing to say which are red, so I didn't. Okay, so, and anyway, it got me the mark. Scientists investigated the role of a protein called KEMP-W in mitosis. The method involved cell fractionation and ultracentrifugation. They began by lysing cells and organelles using a detergent that dissolves lipids in water. Suggest how the detergent releases the CNPW from cells. And that is, of course, they tells you that the detergent dissolves lipids. Well, the one lipid that we know about is phospholipids in the phospholipid bilayer in the cell surface membrane. Therefore, it would rupture the membrane that's going to dissolve them, and it would release the CNPW. I'm going to be honest, I was not sure I'd got the 2 marks here, but I just didn't know what else to write. But I ended up getting the 2 marks. I did write both things in one statement. I don't know why they even wrote that because it's really quite common. The phospholipid, because I said the phospholipids in the cell surface membrane, I think it's pretty obvious that I know that phospholipids are made, cell membranes are made from phospholipids, and it dissolves. So I got the marks. Explain how ultracentrifugation separates CMPW from other molecules. I'm going to be really honest, I was not sure what to write here. So I did go a little bit off on one, and I realised when I read the mark scheme that I didn't actually stick to answering that. So I kind of explained that centrifuging at high speed would cause the organelles to go into the pellet, and if the supernatant containing the lower density CMPW was spun at an even higher speed, the protein would then be separated out in the pellet. So there's my answer. Now, notice that what they were fundamentally saying, how does centrifugation work? Well, it works because stuff is spun at a high speed and it separates stuff out by their different densities. Or you can say mass or size even. Density is the most accurate term. So I think I ended up getting the 2 marks kind of by the skin of my teeth. But because notice that I ended up using the knowledge that I had as best I could and the terminology. So I hope that helps. Now they tell you that this protein is involved in the formation of spindle fibers in mitosis. Spindle fibers are made of tubulin. And they tell you that they treat the cells with small interfering RNA. This causes interference of the expression of the CNPW gene. Worth noting that the gene, when it's written, is always written in italics. And when you talk about the protein, it's always written non-italicized. Don't know if that helps, but it's a scientific convention. The scientists took samples of cells at these times. After adding the siRNA, they then used gel electrophoresis to separate these 2 proteins from these samples. And here are the results. And it tells you that the size of each band is proportional to the amount of protein present. Suggest how the siRNA produced these results. Suggest means this is something that you won't have come across before, but they're asking you, they could have said explain, because they're asking you to apply your knowledge of how siRNA works to how these results were produced. So here am I, as I'm writing this blurb, I'm just reminding myself that siRNAs are these little bits of RNA. Now, I don't know if something's gone wrong. If something has gone wrong on YouTube now with the live stream, can you let me know on the comments? That would be great. The siRNA is this little piece of RNA that when it binds to the mRNA, it breaks the mRNA and therefore the protein won't be produced. Also, if there is nothing wrong on YouTube, can you also put that in the comments? That would be great too. And therefore, here is my answer. The siRNA binds to the mRNA of the gene by complementary base pairing. This causes the enzyme associated with the siRNA, and you don't need to know the name of it, to cut the mRNA with reducing translation of the gene and therefore reducing the amount of protein. As CNPW is involved in spindle fiber formation, as the amount of CNPW reduces, so will the quantity of tubulin. And here are the mark points, and here is the rest of it. And here, this is where I got the marks. Question 6. If you can let me know if something's gone wrong on the channel, that would be fantastic. Thank you. I am just going to check on the screen. Okay, I think everything looks okay. I think I'm going to carry on. Okay, so question 6, ammonia in the soil is oxidized to nitrates and nitrates by species of nitrifying bacteria. Theoretically, you should know that. And then we've got these two soils which contain different communities of nitrifying bacteria. And one soil sample from site A has pH of 6.9, fairly neutral, and one has a pH of 4.3, which is more acidic. And they measured the concentration of ammonia in the soil. So notice the ammonia is going to be disappearing as it's nitrified, as it's turned into nitrates. So they measured the concentration of ammonia in the soil samples over 20 days. They had the same concentration of ammonia at the start, blur. And this is the soil sample B, the acidic soil sample. This is the more neutral soil sample. And this is where they combined the two soil samples together and adjusted the pH to 6.9. And I'm already kind of thinking, okay, the acidic pH, this is going quicker because the amount of ammonia is going down faster in a shorter period of time. This is slower, and this is even slower still. Now, that is kind of already saying to me that at least definitely the bacteria in soil sample B don't like being at 6.9. They prefer 4.3. But of course, the first question is about something completely different. Suggest why these units are used. Well, micrograms, because there's a tiny amount of ammonia in the soil. And per gram, because micrograms per gram literally means micrograms per gram divided by grams, which is why you use the minus sign, because it means it's on the bottom of the fraction. And so grams, there we go, that's what I put for that. And per grams, and this allowed the concentrations of ammonia in different soil samples to be compared. Because if you just use a standard amount of soil, then you can compare different soil samples, because almost certainly when they work it out, the mass of the soil is going to be different in each sample. So hopefully that makes sense. So I ended up getting the marks, mark 0.1 and mark 0.3. I wouldn't have thought of mark 0.2, would never have thought of writing it. Okay. Yay, Munib, nice to see you, and thank you for your little note. That really helps. Okay, another calculation. Calculate the difference in the rate of breakdown of ammonia per day between days 0 and 2 in soil A and soil B. Okay, so it's the difference in the rate, notice, per day, and it's the difference. So you've got to work out the rates for A and B per day between days 1 and 2, and then take one away from the other. So here's day 2. I've worked it out on the graph because you've got 20 little boxes for 5 units. So each unit is 4 boxes. And so this is day 2. And therefore I can read off very easily here that this is 4, and for this is for B, and for A, this is here I've just been silly. That's 2, isn't it? Because of course we've got 10 little boxes for 5 units. So each unit is 2 boxes, and this is 11. So notice the rate of breakdown. You've got to work out how much is broken down. They both started at 20. So the amount broken down in B over 2 days is 18, 20 minus 2. And the amount broken down in A over 2 days is 20 minus 11, which is 9. So here we go, rate of breakdown. Of course, rate, you are dividing what the breakdown by the number of days. So this is the rate of breakdown for B, this is the rate of breakdown for A, and therefore the difference is 4.5. And here are your units, because it's now micrograms per gram of soil per day of time. Okay, hopefully that makes sense. And I got the marks. Okay. 6.3, scientists were nearly at the essay, which I am going to go through. The scientists concluded that the soil mixture experiment showed that there were different communities of soil, different communities of bacteria in soils A and B. Remember that communities means lots of different species. So we've got a whole mixture of species of bacteria in soils A and B. What evidence from figure 6 supports their conclusion? Now, the first thing I noticed that it said supports. I'm not being asked to evaluate. I'm being asked to support the conclusion that there were different communities of bacteria. And I'm also noticing that it says, what evidence from figure 6? So we are now trying to pick out the evidence from this graph to say that there are different communities, different groups of nitrifying bacteria in soil sample A from site A and the soil from site B, which had different pHs. So this is what I wrote. And I first wrote, and I said, because lines A, B, and A plus B have different rates of ammonia breakdown. The rate of soil A is faster than soil B. Or the rate, I should have said, in soil A is faster. But here's the other thing. As I said, as I was looking at the graph in the first place, this is an even slower rate. And that just tells me the bacteria in soil sample A must still be happy because they're at the same pH. But the bacteria that have come from soil sample B will not have been happy because they're going from an acidic pH where they clearly work very well to a more neutral pH where they don't. So I just said this implies the rate of nitrification was slowest of all. This implies that the community of bacteria in soil A were not at their optimal conditions and therefore not nitrifying at their normal rate. So here is the mark scheme and the little pointers, and here are the marks. I ended up, now let me just explain. I clearly got mark 0.1. Oh, sorry, I clearly got mark 0.2, let me say, because A plus B is slowest. I clearly got mark 0.2. For mark 0.1, I had to say that B had the fastest rate of breakdown. Now, I only said, oh, do you know, I got them the wrong way round. I've just realised. I wrote the wrong thing. I would not have got that mark. Let me just correct that. Okay, I'm pleased to looked at that. But if I had written them the right way round, because I've just said the rate of soil in soil B is faster than the rate in soil A, notice that when I then said that A plus B was the slowest of them all, by logical conclusion, I was saying that B was the fastest. So if I had written them the right way round, I would have also got the second mark. And I think I would have got this third mark, suggests that pH 3 doesn't work, or the B soil doesn't work well at pH 6.9. I think I said that in other words. Okay, and now we're given another graph where, let me just remind myself what this is about. It involves the enzyme ammonia monooxygenase, which you've never heard of. Don't panic. Keep breathing. Each species of nitrifying bacteria has its specific AMO-A gene that codes for ammonia monooxygenase. In a second investigation, scientists determined the expression of that gene in the two species S and T. S was from acid soil, the 4.9. So here am I just saying, oh, look, it's the AMO-A enzyme that does this nitrification. S is soil A, the acid one, and T is soil B. They grew cultures separately in soils of different pHs. They determined the amount of mRNA from the AMO-A gene in each culture. And not terribly surprisingly, the AMO-A gene from the 4.3 bacteria went down, the production of it went down as the pH increased. And not surprisingly, it went up for T because it was getting close to its, optimum. Okay, and now I suspect they're going to ask me another completely different question. And I've just said the pH of the soil affects the transcription of the AMO-A gene. Okay, and inevitably, completely different question. In which species was the number of copies of mRNA more affected by changes in soil pH from 4.9 to 7.5? Use a calculation to support your answer. Now, which was more affected? Well, I'm kind of going, okay, we're going from, for S, we're going from 1 times 10 to the 6 to 1 times 10 to the 4. And from here, we're going from 1 times 10 to the 2 to 1 times 10 to the 4. Now, this is about recognizing and understanding almost logarithmic scale. This is a logarithmic scale because it's going up in factors of 10. This is 10, this is 100, this is 1,000, this is 10,000, this is 100,000, and this is 1,000,000. Okay, so therefore, it is not, in my mind, you've got to work out percentage change to work out which has got the greatest change. So I worked out the percentage change for each of them. So for S, 10 to the 6 to 10 to the 4, if you take 10,000 away from 1,000,000, you get 990,000. Over 1,000,000 to work out the percentage decrease, because we are going from a higher number to a smaller number, we are dropping down. We want to know what this difference is as a proportion of the whole original number, this original number here. So you are doing the change divided by the original number, times by 100 to make it a percentage. There was a 99% decrease, which I know doesn't look like it is on a logarithmic scale, but if this was a linear scale where you had 1,000,000 right at the top, 10,000 would be way down near the bottom, very, very close. Whereas T goes from 100 up to 10,000. Well, that difference is 9,900. But here you're going from a small number up to a big number, and you're wanting to see what the percentage, what the difference is as a percentage of the original number here. And it is way over 100%, because 100% would be doubling it. It's 9,900%. So therefore, T has this bigger increase because species S had a 99% decrease, but T had a 9,900 increase. Now, so here's the mark scheme. Here we go. And I got the 2 marks. Okay. If I had just put species T full stop and I hadn't included that, I would hope that examiners would recognize that a downward arrow means a decrease, because I've talked about percentage decrease and this is percentage increase. I think almost certainly, even though I had used abbreviations for increase and decrease, I strongly suspect that I would have got the 2 marks. But notice this got you no marks at all. This method allows scientists to estimate the expression of the AMO-A gene in each culture, but not the growth of the bacterial population in each culture. 4 marks. I have to say, at this point, I kind of went, well, I think I can get 1 mark. I wasn't sure about any others. So the reason I got the 1 mark is because really obviously they couldn't determine the growth of the bacterial population because they didn't measure the number. They had no estimate of the size of the population at the beginning or at the end of the experiment. So clearly they couldn't work out the growth in the bacterial population. And I honestly had no idea what else to write. And then I read the question again, and I kind of thought, how did it allow? Because I had emphasized not the growth in the bacterial population. And then I realized that there was the other aspect of the question. How did this method allow the scientists to estimate the expression of the AMO-A gene? Well, it allowed them to estimate the expression of the AMO-A gene because they were able to count the number of mRNA molecules. At which point, I could not think of anything else to write. So that's all I wrote. So here is the mark scheme. Here are the additional comments. And I got 2 marks. Honestly, I thought I was dredging the bottom of the barrel for this one. I really felt like it. I simply would never have thought of writing mark point 3 or mark point 4. And that is just me being honest. So I wouldn't have got those marks. The scientists set up their cultures in sterile glass bottles, suggest one suitable method for sterilizing the bottles, and explain why it was necessary to sterilize them. So here am I. Now, as a teacher, I would have wrote autoclaving. As a student, I think you're less likely to remember that because that is something that is largely done in the lab preparation area and not in the laboratory itself. Often students don't see an autoclave. So I went down a more student answer for this. So I said they could put them in boiling water. Did I know how long? No, I flipping didn't. So I made it quite a long time. Probably 5 or 10 minutes would have done it to kill bacteria, but let's make sure. So I wrote that. And here, my logic, whenever there is anything about why sterilization is important, my first thought was to kind of go, okay, it's to avoid contamination. And I know that just writing avoiding contamination hardly ever gets you a mark. So, my rule of thumb is to always say where the contamination will have come from and where it would have gone to. So hence, no bacteria in the bottles could contaminate the cultures. Here's the mark scheme. So I got mark point 1, but I don't think I would have got, I don't think I would have got mark point 2. Because I didn't really say that they were removed. I really should have said they were killed because it kills that was daft. And I also didn't extend my answer to say that might break down the ammonia. I just know I don't think I'd ever have done it. So, hey ho, just right. Essay time. I know this has taken a while to get to. So I am now going to go through how I would choose which essay to write. I'm going to go through the basics of how you write a synoptic essay. I have already done a video on the synoptic essay, so I am not going through the whole lot again. But as you know, I'm sure, you're given two essay titles and you have got to obviously write one of them. My tip to you on this, for paper 3, I would always say to students, when you sit down in front of paper 3, I would always immediately go straight to the essay question. I would then really quickly scrawl, and I'm going to show you what I mean by scrawl, an essay plan for the 2 essays, taking not much more than 5 minutes, maybe really less than that, preferably. And on the basis of those essay plans, you should have come away with an idea of which essay you're going to do. I would then go back to the start of the paper and do the rest of the paper, because the rest of the paper might jog your memory of something you could include in the essay, particularly any extra to syllabus stuff, but also it also gives your mind, just even subconsciously, you know, just aware that those questions are coming up. You'd be thinking about it even as you are turning back the pages. So there's my logic. So let me just firstly go through the 2 parts of these titles. The bit I've highlighted in yellow is fundamentally the topic of the essay. It's the subject of the essay. And I've particularly highlighted that it is in cells and in organisms, and here it is between cells and between organisms. So just notice those words, read the essay title carefully so that you've visualized it properly in your mind. That's the subject or the topic of the essay. The importance of, and it's clearly the same in both of these, is the theme of the essay. So, worth noting that the topic of the essay are the AO1 marks of the essay. I said that most of the AO1 marks in this paper is in this essay, and they are. 15 of the marks out of the 78 on this paper are in this essay. That means, can you remember knowledge of 4 or 5 examples of control of movement in either cells or in organisms? That's your basic thought. Can I think of 4 or 5 examples of control of movement in cells or in organisms, and can I describe them? Can I regurgitate the knowledge? Because you're basically wanting to write 4 or 5 paragraphs to answer this essay. So 4 or 5 paragraphs, each paragraph to include, each paragraph to include, number 1, a description of an example of the topic. So each paragraph is has a description of the example of the topic. AO1 marks. What you need to then include in that paragraph, and you can absolutely tag it on at the end of your AO1 description, is then talk about the importance. Let me say that again. Then you can relate that content that you've just written for that example. Can you relate it to the theme of the essay? In other words, can you talk about the importance of the example that you've just written about. So, hence I've written, you also need to include in each paragraph sentences relating the example, what you've just described, to the theme. In other words, what is the importance of the example? Hope that's clear. So you can gain 15, notice the majority of the marks, 15 out of 25 of the marks, just by writing 4 or 5 different decent descriptions. To get the higher marks, 16 through to 25, you've got to nail writing about the importance of each of your examples. I say 4 or 5 paragraphs because examiners take the best 4. So if one of them ends up being a little bit dodgy, they'll take the best 4 to base your mark on. 5 is kind of hedging your bets. If you haven't got much time, for goodness sake, only concentrate on writing 4 and write the 4 best ones you can. But notice the 4 best paragraphs are going to be the 4 that you can write the best description and the best importance of bit. Hopefully that makes sense. A word on getting the highest grade. Can I just say, whenever I was teaching in school, even to my A-star students, I used to say to the whole class, which was a mixed class, I used to say, I am going to teach you how you can write the best essay for your ability. I'm actually not going to emphasize getting the highest possible marks, the gold standard 24 or 25 marks out of 25. Because to do that, you have to both. Now, notice this one, because everybody seems to forget this one. You have got to write an amazing essay up to the standard of 23 out of 25. This is an A-star essay as it is. And on top of that, you have got to include extracurricular knowledge. Now, I'm going to be really pragmatic about this. If you are a C or a B grade student, basically, I'm going to be honest, anything lower than an A, or probably anything lower than an A-star, there's no point in you, if you write a perfectly reasonable essay, but not an absolutely perfect one, and you include extracurricular content, that isn't going to jump you up to a 24, 25 mark essay, because you've got to have nailed writing a flipping brilliant essay anyway. So my emphasis is getting you to write a good essay for the level that you are aiming at. So, A-star, you're looking to kind of, if you're really wanting to write an A-star essay, you're looking to get, I think, 22, 23 plus marks out of 25. For an A, you're looking to get about 20. For a B, you're looking for about 17, 18. For a C, you're looking for about 15. For a D, you're looking for about 12, 13. And for an E, you're looking for about 10 out of 25. That's for your essay to reflect your grade that you're aiming for. And I generally say to students, let's not get too hung up. I'm not capping your ability, but let's not get too hung up if you are only writing an essay to your grade level of ability. Please notice that is not me restricting students. I'm just trying to be pragmatic. I am always delighted if students end up writing a better essay than I would have expected. Yeah, hopefully that's clear. So here am I planning the 2 essays. All right, remembering the whole. Now, there's the important thing, not only thinking about the topics, the examples of the topics that I know about, but also whether I can write about their importance. So here is my really quick spider diagram, literally 5 branches of it, movement in, and I've literally, but it's actually control of movement. And that's where I started. I'm kind of thinking control of movement. So I kind of, okay, muscle contraction, neuromuscular junction, calcium ion release, synapses. I thought control of water potential because in the kidney, because of course water is being reabsorbed, it's moving. Synaptic control, that would be, sorry, excuse me, that would absolutely, your whole summation stuff, that would be control of movement. Control of muscle contraction. Here I was going to talk about the neuromuscular junction and calcium ions. Control of heart rate, because of course that is control of movement of the blood. And also diffusion, osmosis and active transport, because the control there is by the channel and the carrier proteins. But I must admit, I kind of thought, even content-wise, I'm kind of going, oh, you know, that felt harder. So interactions between cells and organisms, I came up with my 5 much more easily in terms of the content that I could write about, describing the examples. But then I started, and so I would have naturally have gone for 7.2, but then I did always, don't just stop there, always then think about, but which of these examples could I talk about the importance of them most easily? And my guide for talking about the importance of something, in the examiner guidelines, the importance has got to be talked about to A-level standard. That means you've got to use A-level terminology and A-level content. So my guide for writing about the importance of an example is to try and relate the example that you've just written about to some other topic in your A-level content, because then of course you have got a mine of terminology that you can get into your answer. If you're using A-level terminology, you're writing to A-level standard. That's pretty obvious. And all of these topics, and basically the importance of, you're either thinking about, well, what happens if this example, what would happen if the nitrogen cycle isn't there? Or thinking about the real usefulness of the nitrogen cycle. And that's what you're almost thinking about in both cases, every time, for each of your examples, you're thinking about why does this example benefit biology, or the organism? Or what would happen if it wasn't there? Good ways to think about it, but then try and link it to another topic. So this is what I've done here. So I'm ticking this one because I think I can nail the importance more easily. So here, nitrogen cycle, I know that I can go into the whole thing about nitrogen's needed to make DNA and proteins, and therefore I can bring in, and therefore it allows DNA replication and cell division, or proteins, enzymes, you're suddenly mining another part of your knowledge to A-level standard. So here I thought energy transfer, energy transfer up ecosystems. Why do organisms eat other things? Well, that's because they need energy, so that respiration can happen in their cells, and muscle contraction. I'm kind of going to go down that route. Here, the nerves and synapses, that's my abbreviation here. This is about an organism being able to respond to the environment. And, and therefore, if it can respond better, natural selection. And estrogen and adrenaline, the whole secondary messenger thing, again, I was kind of going down the line of responding to the environment. Oh, sorry. And I didn't put in one for immunity, and I'm now thinking about what I did write about. I think, and here and there I was kind of going down, you know, being able to resist infections, being immune to them, and therefore surviving. But notice the thing that students tend to do when they're talking about the importance, they kind of said, oh, well, if this didn't happen, the organism wouldn't grow. And I'm kind of going, that isn't A-level standard. It might be true, but it's not A-level standard. And that's why I'm saying, try and mine another topic in your A-level to use the terminology from another topic, because then you know you're pretty sure you're talking to A-level standard about the importance, even if you do end up saying, oh, and therefore it won't survive. Can you see where I'm going with this? Hopefully. So here is my essay in note form. These are bullet points that basically each one of them would be at least probably a sentence, but notice equally that it isn't huge numbers of sentences, because it doesn't have to be huge numbers of sentences, but equally notice that every sentence is going to be full of A-level content and A-level terminology. There's the crucial thing. So this bit in dark blue is my content, my A01. So my first one I'm going to talk about is the nitrogen cycle. So first thing I would say is that it allows cycling of a finite resource, nitrogen. Nitrogen, if it wasn't recycled, we would run out. Yeah. Hey, that could have come into the importance. That could be an importance thing. Then I start. Now, this has got to be where it's got to be brief. Don't get to go into every bit of detail, but think about parts of the cycle that you can talk about and get the terminology in without spending the whole 30 minutes or 35 minutes that you should take to write this essay on writing one paragraph. So I think I would say nitrogen fixation occurs as a result of the interaction of mutualistic bacteria in the roots nodules of leguminous plants. Interaction between the plant, or mutualism being an interaction. Then I would say that then other bacteria nitrify the ammonia from nitrogen fixation into nitrates and then nitrites. And that the interaction of the bacteria allows the production of nitrates, which plants can then absorb. Plants are then eaten by primary consumers, and secondary consumers eat the primary consumers. The nitrogen is passed up the food chain. It's how nitrogen moves up the food chain. But all organisms die, and animals certainly produce waste, feces and urine. And these are ammonified by saprobionts back into ammonia. I haven't talked about denitrification. I'm not too worried. There's enough content there for the paragraph. And here's my importance bit. The importance, nitrogen, the accumulation of nitrogen in an organism allows the synthesis of DNA, amino acids, brackets proteins, and ATP. Those are probably the three most important molecules. Also includes coenzymes, NAD and NADPH, but you might, or NAD and NADP, but you might not remember those two. So this enables growth of the organism. This is important because it enables growth of the organism. Now notice, I haven't just finished there. I'm now going to launch into my A-level standard via DNA replication using DNA helicase and DNA polymerase plus mitosis to bring about that cell division. Yeah, you could put cell division here. It also enables enzyme synthesis because all amino acids have nitrogen in them. And the importance of enzymes, the synthesis of enzymes is really important because they lower the activation energy of reactions, and that allows reactions to happen at the low temperatures that occur in all organisms on this planet. So that allowing growth and survival. Notice that I have basically just said this allows organisms to grow and this allows organisms to survive. But I've brought in other bits of my A-level content to provide me with the terminology to talk about it to an A-level standard. So, second paragraph, and notice I have not written a introduction or a conclusion. Don't need to. Sorry, I've just realised. Munib, I am absolutely delighted with that little comment. Thank you so much. And Sian, I completely understand that as well. Okay, paragraph 2, energy transfer. So here is my knowledge bit, AO1. Plants synthesize their own food. That's me. Now that's almost GCSE, brackets, glucose, yeah, via photosynthesis. You could put via the light independent and the light dependent reactions, yeah, which, you know, allows the plant to absorb light and fix carbon dioxide. You know, show them that you know a little bit. You don't have to describe the whole thing. Don't describe the whole thing. That would probably be close to irrelevant. You know, you're going off on one. These are, because the focus of your thing is transfer, or the focus of my paragraph is transfer. These are eaten by primary consumers, which leads to digestion and absorption, because that's the only way that they'd get the nutrients into their body, to provide biomass for respiration and for growth. Okay, because the amount of stuff, the amount of food eaten, that goes into increasing, here am I talking about me, increasing my biomass, but obviously any of that biomass that is used by my cells in respiration isn't going to go into growing my body, making me bigger. And dare I say it, any of that biomass that I eat that ends up going through me, now either egestion or as a result of me producing urine, those things also include energy, that isn't going to go into me making my biomass as well. So, you know, which is why it provides biomass for respiration and for growth. Primary consumers are predated. So talking about predation, and you could absolutely talk about the predator-prey cycle. Here am I just describing it really briefly, saying that the prey numbers increase and decrease before the predator numbers. I'm not going to go into a huge amount of detail, but I'm showing the examiner that I know A-level content about energy transfer. Here's my importance. So I've started down here and I've gone up here. The importance is, I can't see below the little box. Energy change required. Energy transfers are provided. I can't read what that bit at bottom is. I can't remember. Energy transfer is required by all, or energy is required by intake is required by all organisms, because the transfer is linear up a food chain. The sun is the constant source. It is the source of energy on the planet. That goes, allows the plants to fix carbon. Yeah. Sun drives the food chains. And I have said eating, digestion and absorption is the only way that glucose is provided for respiration. Here we go. So you notice that I'm bringing in another topic, which as a result of glycolysis, link reaction, Krebs and oxidative phosphorylation, produces ATP for chemical reactions and for muscle contraction. Again, notice that I'm bringing in another topic of my A-level biology to mine the A-level terminology. And, you know, I could say that energy is absolutely crucially important so that organisms can move in response to their environment. For example, a prey animal running away from a predator. Paragraph 3, neurons and synapses. So I start off by saying that neurons is the content. Neurons are specialized cells which conduct electrical impulses from receptors to the CNS. Those are the sensory neurons, and from the CNS to the effectors. And I think I would say these are muscles and glands, and those are the motor neurons. The CNS is full of relay neurons. Receptors detect stimuli. They are specific for each stimulus. And action potentials are how nerves conduct the electrical impulses down the neuron, and these are reversal of charge across the membrane. Synapses are junctions between neurons, and this is where decisions are made about whether an action potential continues down the postsynaptic neuron. Notice that when I'm talking about it, I'm going to be bringing in that A-level terminology. And those decisions occur via temporal and spatial summation. Those are the key ones that you've learned about in your A-level syllabus. And if you've got time, you could possibly describe those. And again, the neuromuscular junction. So here we are talking about interactions between cells. Here we have the interaction between the motor neuron and the muscle fiber. And can I just say, throughout this essay, I probably would be just emphasizing, oh, and here we have another interaction between organisms. Oh, and here's the interaction between this cell and that cell, for example. The neuromuscular junction is at the end of the motor neuron, which allows the impulse to reach the muscle effectors, the muscle fibers. And therefore, I've talked about quite a lot in that whole topic. Here's the importance. Animals must be able to respond to their environment to avoid danger, for example, predation or injury, and also to feed. And I'm just going to point out, both herbivores have got to find, so monkeys that only live off fruit have got to be able to identify the fruit by the stimulus of their eyes. Predators have got to be able to go after their prey. And in some cases, care for the young. Now, why did that come in? I don't know why, the way I was thinking. So they've got to be able to, the whole idea of organisms, the whole raison d'etre of all organisms in biology is to, from a biological point of view, not social, from a human point of view, the whole biological raison d'etre is to pass on genes to the next generation, pass on those advantageous alleles. So hence I've talked about the young. So effective responses to stimuli increase the survival and successful reproduction of organisms, leading, I could have said, to the greater chance of advantageous genes being passed down and natural selection happening. Paragraph 4. Estrogen. Now, at this point, I decided I was only going to write about estrogen and not about adrenaline and the secondary messengers, just decided. So estrogen is a sex hormone. It increases the thickness of the uterus lining. To be honest, that is GCSE content. Ready for implantation of a zygote or embryo. You could write either. Estrogen is lipid soluble. I'm now hitting A-level content, and therefore passes through the cell surface membrane of every cell, because it does. It goes straight through the phospholipid bilayer as a result of diffusion. because it's lipid soluble. There's nothing stopping it going from a higher concentration in the blood or in the tissue fluid to the lower concentration in the cell. And therefore, receptors are required on the inside of the target cell, i.e. the uterine lining, for the effect of the hormone to come about. So the receptor in the target cells of the uterine lining are on the transcription factor. This is now you talking about how estrogen works. On the transcription factor, and when estrogen binds to the receptor on the transcription factor, the tertiary structure changes and it allows the transcription factor to move from the cytoplasm into the nucleus and bind with the promoter on the DNA, bind with the promoter on the DNA, which I'm going on here, here we go, so that mitosis, now here am I noticing that this is for uterus lining proliferation. That, in my A-level knowledge, means that mitosis is, if a lining is getting thicker, it's kind of logical that more mitosis has got to happen. The gene for mitosis on DNA. This is interaction between cells in the ovary and the uterus. Because the ovary is where the estrogen is produced, the uterus lining is where its target organ. So, importance. Without the increased thickening of the uterus lining, implantation would not occur and therefore no successful reproduction. Advantageous alleles, any advantageous alleles in that individual could not be passed on if reproduction was not successful, leading to the individual's alleles which decrease reproductive success, i.e. the disadvantageous alleles, decreasing in the population. And that is the definition. In fact, I've just got that wrong. That is the definition of evolution. But that evolution has occurred by natural selection. So I'm hoping that makes sense. Notice I'm bringing in another topic. And finally, paragraph 5. I don't know about you, I am exhausted. Paragraph 5, this one is on immunity. This is the interaction between cells of non-specific and the specific immune response. I'm nailing it right at the beginning. And in honesty, I should have done that I should have introduced each of my other previous paragraphs, just kind of going, why am I using this example? Well, because this shows interaction between organisms, or this shows interactions between specific cells. Anyway, so here am I now kind of going, oh my goodness, how do I describe the immune system in a few sentences? Notice how jam-packed every sentence is going to be of A-level content and terminology. Phagocytes, maybe not do, maybe you could say carry out. Phagocytes carry out phagocytosis. And once they have digested and absorbed the pathogen, they present the antigen on their cell surface membrane. Binding of the antigen with the T helper cell brings about clonal selection and expansion, which gives rise to T killer, T helper, and T memory cells. The T killer cells kill infected host cells. Notice more cell-to-cell interaction. This is the cell-mediated response. The T memory cells stay in the blood, just in case the same infection occurs again. T helper cells bind with the B cells, notice cell-to-cell interaction, and the antigen on the pathogen to bring about the clonal selection and clonal expansion of the B cell. This gives rise to plasma cells and B memory cells. Plasma cells secrete antibodies, which bring about the destruction of the pathogen throughout the body, the humoral response. And the B memory cells stay in the blood. That is 1, 2, 3, 4, 5, 6, 7. 7 jam-packed statements. Now I think I can now write something about the importance. The T killer cells interact with the host cells. Do you know? Yeah. Cells containing the pathogens being killed, and therefore the pathogens being destroyed. Plasma cells, the plasma cells' antibodies cause the destruction of pathogens in blood, lymph, and tissue fluid. Effective cell-to-cell interaction leads to an individual recovering from infection. Yeah, this increases their chances of survival from the infection, and it increases their chances of reproduction, alleles being passed on. The memory cells, B memory cells and the T memory cells, cause immunity. And this means that this individual is even less likely to die from the infection, even if the infection comes around again. This again increases the chances of the organism's survival and ability to reproduce, being able to pass on those advantageous alleles and the allele frequency of them increasing, giving rise to, and I should have put here, evolution by natural selection. That is what I would have written. What mark do I think I would have got? Do you know, I think 21, 22, possibly 23. Not 24, 25. I haven't included anything from outside the syllabus. But I think all of my descriptions would have been packed with A-level terminology, and I think that I would have been able to nail showing how the content is related to the theme of the essay, the importance of those interactions between the cells, and doing that to A-level standard. Can I just write a note where the exam board talks about the paragraphs have to be interrelated? They are talking about the interrelation between the content, the description of your example, and relating that to the theme. They're talking about this importance bit. You do not need to be, for an A-level examiner marking your essay, you do not need to connect your 4 or 5 paragraphs, because some of those links would be tenuous to the point of ridiculous. This is not supposed to be a beautiful essay. It doesn't have an introduction, it doesn't need a conclusion, and it doesn't need for the actual examiner to mark them. It doesn't need the paragraphs to be linked seamlessly. What you do have to nail is nailing the description, the AO1 marks, and nailing those AO2 marks by talking about the importance of each example to an A-level standard. And most students really struggle with either one of those two things. And if you can do even the AO1 really well, do those descriptions, you have the potential of getting up to 15 out of 25. That's a good C grade mark. If you can nail at least the importance for a few of your, let's say 2 or 3 of your paragraphs, you're then kind of justifiably expecting to go up between B and an A. So that's kind of how it works. I hope that has been really helpful